We are very much happy to welcome Professor Rachid Thiam for his talk on the interface of biophysics and membrane biology. Rachid was trained in physical chemistry and microfluidics at ESPCI in Paris and got interested in lipid metabolism during his stay as a graduate fellow at the Department of Cell Biology at Yale University in 2011. 4 years later, Rachid returned to Paris and established his group at CNRS and the Laboratory of Physical Statistics in ENS for the development of multidisciplinary approaches combining soft matter biophysics with cell biology. As a young investigator, he received the Claudie Paoletti Prize in 2018 for his outstanding research in the field of biology. His team uses synthetic model systems to understand how membrane properties affect the behavior and dynamics of lipid droplets, as well as their functionalization through partitioning of proteins from the ER to the lipid droplet monolayer. Rachid, we are all ready to listen to your talk.
Okay, thank you, Katerine, for this nice introduction, and I thank the organizers for inviting me to this nice symposium. It's quite unique symposium, I have to say. So do you hearing? Are you hearing me well? Hello.
Yes, we can
Okay,
hear you
great.
well.
Okay, so let's start. So maybe we can start by this classic of cooking, right? And it's about how to make a mayonnaise. So how do you make a mayonnaise? So you need an egg, right? You need an aqueous phase, and you need an oil phase. And if you shake these components, you end up getting a mayonnaise, which looks like this. And then if you would zoom in the mayonnaise, what you would have is basically droplets, many droplets that are packed like this. And these droplets are actually oil in water droplets, called an oil in water emulsion droplets. And this works because at each of these droplets, you have a phospholipid monolayer and proteins. And that's why when you make mayonnaise, you have to take the yolk, not the white. Why? Because to have a good mayonnaise, you need to balance the oil to phospholipid ratio, and you need to balance the phospholipid to protein ratio. If you don't do so, let's say, for instance, you have less phospholipids than, or if you change this ratio basically, and you're having more proteins, typically when you have more of the white, what you get is not a mayonnaise. You get another type of dressings. And you may want these dressings, but these are not mayonnaises. And what are these dressings? These are basically also water aqueous phase with large droplets or phase separated phases. And how you reach these phases, basically you have droplets that merge, as you can see here, to give larger droplets. And if you continue merging these droplets, you end up having this phase separation. And it's been a century of work that yielded a good understanding of how droplets are stabilized, like in this case, or destabilized in this case. And this has been a focus of research from food to medicine. And these are crucial for generating any type of product you have currently in the market. So 10 years ago, I was amazed when I discovered and I came to meet the most ancient droplet maker, the most ancient emulsion maker, and it is the cell. So cells are capable of making droplets of specific features. So here you have lipid droplets in a liver cell. So these are basically oil in water droplets as previously. So structurally, each of these droplets is made of an oil core containing neutral lipids. These neutral lipids are in general triacyl glycerols or cholesterol esters. Also, like mayonnaise droplets, they have a phospholipid monolayer, and they have also proteins. And these lipid droplets play a crucial role in the cell. So here I'm showing you what I just described, for instance, a lipid droplet with a protein that is labeled at the surface. So what I was saying is that these lipid droplets play a crucial role primarily in lipid metabolism. So they regulate the energy metabolisms of cells. They regulate the lipid levels and type in cells. And what is interesting is depending on the cell type and metabolic function, the droplets can be diverse. They can be diverse in their oil content. They can be diverse in size, like here in adipocytes. They can have different protein at their surface, and they can experience different phases. Some droplets can be growing, others can be shrinking. And when I came up to know all these exciting questions of lipid droplet biology, I decided to take advantage of our knowledge of emulsion science, so droplet science, to understand how lipid droplets are regulated in the cell, to understand lipid droplet biology basically. So one of the most and current important question of lipid droplet biology, for instance, is how lipid droplets are formed in the cell. So lipid droplets are, Pedro just mentioned, are formed at the ER membrane. So here in green is the ER, and in red are lipid droplets that are being formed at the ER. And you can appreciate that in this magnification. So concretely, what happens? You have enzymes that sit at the ER membrane that catalyze the formation, the synthesis of neutral lipids, in majority triglycerides, but you have many other neutral lipids that can be made depending on the cell type. So as the concentration of the neutral lipid to the phospholipids increases, these neutral lipids tend to make clusters, more and more clusters, and larger clusters. And when a critical concentration is reached, a process called nucleation occurs, whereby the neutral lipids condense, as in phase separation processes, to form an oil lens within the bilayer. And such oil lens has been visualized by EM in yeast. So here you have the oil lens, the ER bilayer, here you have the ER lumen. And when the concentration of oil keeps increasing, the droplets buds off, forming a spherical droplet embedded with proteins, and which can be connected or disconnected from the ER. So this is really a simplified view of lipid droplet formation steps, but it is much more complicated than this. But for the sake of this talk, I use this simplification. So what is interesting is the way these droplets form depend on the neutral lipid type. So for instance, if you take two types of neutral lipids, one may nucleate at this concentration and the other at this one, and they may require different phospholipids and different proteins. But the neutral lipid also may determine how proteins are targeted to lipid droplets, which is the focus of my talk today. So I will basically try to show you how we divert the emulsion science to bring understanding of how proteins can target to lipid droplets. So there is a first class of proteins that bind to lipid droplets. They are listed here, and this class this is the class I term soluble proteins. So basically, these proteins are in general soluble in the cytosol, and they can associate with the lipid droplet surface. And how does that happen? Most of these proteins bear what's antipathic helices. So these helices actually they are unfolded in solution, and they can fold at the lipid droplet surface to bind the lipid droplet. And what we have shown recently, which was an important question, which was how this protein, for instance, is capable of detecting the lipid droplet surface instead of the bilayer, because it has the choice to bind either to the droplet or the bilayer. And what we've found was that actually the phospholipid density at the bilayer is much tighter than at the monolayer. So for 100 phospholipids at the bilayer, you would have maybe 87 to 90 phospholipids at the droplet. So here, this is somehow a kind of top view of the bilayer surface. Each circle is a phospholipid head group. And here, it is the droplet surface. So the lipid droplet surface is sparser than the bilayer, and therefore an aliphatic helices cis has more accessibility to this hydrophobic region. What was also interesting was that, as I mentioned to you, droplets may have different oil content. They can have different natural lipids. And if you are like a aliphatic helices, you may see the same number of phospholipids, the same packing of phospholipids, but you want to be seeing the same hydrophobicity. And therefore, the hydrophobicity plays a role in how aliphatic helices are incorporated to the lipid droplet monolayer. And this might explain why perlipin 2, which is one of the major lipid droplet proteins, when cells make lipid droplets that are rich in triglycerides, perlipin 2 associates almost with all these triglyceride droplets, while when these droplets contain sterile esters, another type of neutral lipids, which is here in red, perlipin 2 fails to efficiently bind to these sterile ester droplets, but binds these triglyceride droplets. And this basically reveals that the affinity between the neutral lipid and the aliphatic helix is crucial to the association. And we've demonstrated that in vitro. So basically, we've generated, for instance, a squalene droplet. Squalene is a derivative of sterile ester, and a triolene droplet within the same medium, and we've added the aliphatic helices derived from perlipin 1, another lipid droplet protein. And what you can appreciate here is that the aliphatic helix better binds to the triolene surface than to the squalene surface, demonstrating that aliphatic helices can detect the nature of the neutral lipid. And in a nutshell, what we've concluded is that two lipid droplets may have the same phospholipid coverage, but what dictates the recruitment level of an aliphatic helices is the affinity of the aliphatic helices with the oil. If the affinity is low, you have less recruitment. If the affinity with the oil is higher, then you have more recruitment to the monolayer. So there is another class of proteins that associate with lipid droplets that Pedro just described, and which I will spend the rest of my talk talking about. So this is this concerns the monotopic class. Here are examples of proteins belonging to this class, and basically this class of proteins move from the ER bilayer to the lipid droplet surface via ER lipid droplet connections, like here. And they usually bear hairpin motifs or hydrophobic transmembrane helices. So currently there is not a clear understanding what is the preference of binding of this class of protein between the ER to the droplet. In other way, how do these type of proteins partition between the ER and the lipid droplet surface? And we formulated this question in terms of energetical, in an energetical standpoint. Basically, when a protein moves from the bilayer to the monolayer, it is in contact with a different environment, which means there is some energy variation between the monolayer and the bilayer, and we wanted to have an estimation what is this energy gap. So experimentally, what we did was to develop a new assay where basically what we do is we express our protein of interest in a cell. So the protein binds to the cellular lipid droplets here. Then we release the cell, we recover the lipid droplet fraction. So like here, so these are the purified lipid droplets in water. Then we add exogenous triglyceride, exogenous oil. And basically, if you shake this mixture, you end up with having water in oil droplets, which encapsulate the purified lipid droplets. So this is like an inverted droplet. And if you mix this, what happens is these lipid droplets deliver all their protein content to this new interface. And in the meantime, you can add phospholipids to cover this interface. So this approach enables us to have lipid droplet proteins at this new interface, which is more controllable. Then what we do is we take two of these droplets and we can pair them to make a bilayer. So we have two monolayers that are sticking together to make a bilayer. So as you can appreciate in this movie, we have two water in oil droplets covered each by a phospholipid monolayer, and they adhere here to make a bilayer. And in this system, we can study now how this protein, which is initially on the monolayer, partition between the monolayer here and the bilayer there. And we can quantify partitioning by fluorescence. So here we have a fluorescent protein. We can make plots. We can extract the profiles, and we can quantify how much of the protein is on the bilayer compared to the monolayers. And we define this partitioning coefficient. So first of all, what we did was just, okay, let's just try to work with a soluble class, which is the first class of protein I introduced. So when you use majorly PE phospholipids, so phosphatidyl ethanol amine phospholipids, and we worked with this GMAT, which has enhanced hepatic helices known to detect packing defects. So in PE, we have a partitioning coefficient of almost 1, which means this protein doesn't differentiate between the bilayer and the monolayer. But when we change the composition and have it like PCPE 1:1, then the partitioning is largely decreased. So the protein got more enriched to the monolayer side. So what this shows is just like by changing the phospholipid composition, the partitioning degree can be changed. So we've done the same experiment for this monotypic class. We use ACSL3, which is a lipid droplet proteins that resides in the ER. In PE, actually it prefers to the monolayer, and in PCPE, it is almost excluded from the bilayer and largely accumulates on the monolayer, like as you can see here. And with this assay, we've tested many soluble and monotypic membrane proteins. So in green are the soluble ones, monotypic membrane proteins in pink ones. And what you can see is, on average, they all accumulate onto the monolayer, suggesting that proteins by de facto prefer associating with the lipid droplet monolayer. And the other information that was also striking was that if you look at the pink data, they seem to be on average above the green data, meaning that monotypic membrane proteins better partition to the monolayer, to the lipid droplet surface than even soluble proteins. And how we understand that, how we translate that into energetical terms. So basically, when this monotypic membrane protein is on the bilayer, it has a high energy state. And when it moves to the monolayer, it has a low energy state. And therefore, moving from the bilayer to the monolayer is something spontaneous. So all proteins in the ER would tend, monotypic membrane proteins would tend to accumulate to the lipid droplet surface. But this is somehow provocative because that's clearly not the case. And clearly, why? Simply because there must be some kind of regulation. So basically, this direct translocation should be prevented. And somehow the ER puts there some mechanisms to have an energy barrier to go to the monolayer, so that when a protein is at the bilayer, it would have to climb this energy hill. So you need to put some effort to bring it to the monolayer. Okay, so this finding was interesting, but still we wanted to have a more clear understanding of why these proteins would accumulate to the droplet surface, and especially because we've been working with, let's say, genuine proteins, which means what? Which means that they might have multiple binding domains, as here for oleurosin 1, it has a hairpin, but also it has hepatic helices, and most hepatic helices usually are soluble proteins. So it is coupling many binding modes. And also we have protein-protein interactions that are in the system that we don't control. And this is why we wanted to have a more controlled system and simplified the question by asking, how would a simple transmembrane domain, a transmembrane helix, partition between a bilayer and a monolayer? And to address this question, we've worked with a model peptide called QALP20. So it is a leucine-alanine repeat motif. And this QALP peptide has been used to model the transmembrane helix of many proteins residing in the Golgi, in the ER, at the plasma membrane. So we saw that, okay, this is just like a basic hydrophobic sequence. And so if we are able to understand how this basic motif would partition between a bilayer and a monolayer, we would reveal some ingredients that control such partitioning. So first of all, what we did was just to have QALP in the in the system. Here we had a bilayer made essentially made with a lot of PE. Here you can appreciate the phospholipid signal. And what was surprising to us is this peptide was almost absent from the bilayer, right? And what was surprising, we said, okay, maybe the protein wasn't like diffusive and so on and so forth. But then we did some FRAP experiments. So we bleached this area to look at the recovery, and we saw recovery. So you have a protein that is mobile at this interface, but cannot enter or inefficiently into the bilayer, right? So so what this suggests is that QALP, which was initially on the droplet monolayer. So I remind you the system. So we have the protein on the droplet, and then we pair them to make the bilayer. So we work on a system where the peptide is already on the monolayer, and it inefficiently go to the bilayer. And we can understand this because if the energy state is lower here, it would cost a lot of energy to go there. But the real question is whether a protein that is on the bilayer would go onto the monolayer spontaneously, which is in general the right topology. So to address that, we've switched systems. So we've used these droplet embedded vesicles. So basically, we take a giant bilayer vesicle, we add oil droplets, and we have the droplets incorporated within the bilayer, as shown here. Then what we did is we prepare now the bilayer with QALP 20, the peptide. So you can see the signal here. Then we added the artificial droplets. And what we saw was striking was now you see the signal of the bilayer is much lower than the signal on the droplet here. And this confirmed that actually going from the monolayer to the bilayer is not favorable, but going from the bilayer to the monolayer is spontaneous and energetically favorable, as you can appreciate here. Then we wanted to get more insights on why the monolayer is preferred. And what we suspected was that the phospholipid composition would modulate that or is involved. So here is the previous example that I showed you. The helix is not in the bilayer, in PE. Now, if you increase the amount of PC in the system, then it starts to relocalize a little bit to the bilayer. And even more if you start increasing PC. So somehow the partitioning between the bilayer and the monolayer is determined by the PC to PE ratio. And we've done that, we've changing continuously PC-PE, and as the PC concentration increases, we get more into the bilayer, but still we have more protein on the monolayer. And this can be seen in dynamics. So we start with a system where we have a lot of PE. The protein is not on the bilayer. Then we inject PC into the system. And as PC gets recruited to the system, the peptide starts to move into the bilayer. So it moves from the monolayer to the bilayer, like here. And confirming that the recruitment of PC somehow promotes a little bit more relocalization into the bilayer. And on an energetical standpoint, how we can understand that is simply like in the previous situations, this would be the energy gap between the peptide being at the bilayer or at the monolayer. Now, if you increase the PC to PE ratio, you decrease this energy gap and makes the probability for going from the monolayer to the bilayer higher. So you get a little bit more proteins to the bilayer. And one question then is, what happens at the molecular level? How can like the PC to PE ratio control the bilayer to monolayer partitioning? And because we use DOPC and DOPC, DOPE and DOPC, so they have the same acyl chain, right? So no difference in the hydrophobic region that is supposed to be in contact with with proteins. But these two phospholipids differ in head. So DOP has a smaller head than DOP has a smaller head than DOPC. So they have different intrinsic curvatures. PE is conical, while PC would be cylindrical. And now, if you start thinking about thermodynamics, actually you can put in this box different interactions. You have interactions of the peptide with phospholipids, interactions of the peptide with the oil phase, interactions of the peptide with the aqueous phase. So if you have more PE, you have more defects at the monolayer, more packing defects at the monolayer. So basically, you have more interaction between the peptide and the oil phase. Now, if you use more PC, you have a better packed monolayer. So you increase the phospholipid packing level. Therefore, you increase the contact of the peptide with the phospholipids and less with the oil. What does this mean? In this case, the peptide is not anymore capable of distinguishing a monolayer of a droplet and a monolayer of a bilayer because it is experiencing more interactions with phospholipids, which you can find on the bilayer and on the monolayer. So let's assume that this interaction is more favorable. The interaction with the oil is more favorable. And then comes this one and that one. What does that mean? When the peptide is on the bilayer or the protein, it is experiencing interaction with phospholipids from this monolayer and that monolayer. So these two causes. And it might experience interaction with water. When it moves to the monolayer, it is experiencing interaction with the oil phase throughout its length. So you have these green causes. So you have a driving force toward the monolayer. Now, if you increase the phospholipid packing, what happens is you increase the interactions with phospholipids, you decrease the interactions with the oil phase, and then you decrease the K on divided by K off. So if this model is right, if we swap the preferences, then we should change the localization. We should change the partitioning. And how can we do that? We can do that by changing actually the oil phase. And as I mentioned to you, lipid droplets may have different neutral lipid content. But here, for the sake of demonstration, we chose a non-physiological oil so as to generate the worst, let's say, interaction possible. So first, here is the triglyceride oil. So this is the previous situation where the peptide is absent from the bilayer. Now, if you switch from triglyceride to silicone oil, which is non-physiological, what you can see now is that the peptide forms clusters with phospholipids, and now it goes to the bilayer. So how we explain this is that now the peptide wants to be in contact with phospholipids than with silicone oil, so it clusters. So it is enriched in regions where you have more phospholipids, which are basically the bilayer and these phospholipid clusters. And this was a proof of principle, at least to show, to suggest that the interactions of the hydrophobic binding domain of proteins with triglycerides is the driving force for the accumulation, the strong accumulations of proteins, of monotopic membrane proteins. So to summarize, what I've tried to explain to you is that when a monotopic protein, membrane protein, is at the bilayer interface, the system has a high energy, but when it moves to the monolayer, it lowers this energy. And therefore, this transition is something spontaneous. But the cell doesn't want that. Otherwise, lipid droplets would somehow suck off all the proteins, or like retrieve all proteins that are in the bilayer. And to prevent that, there must be regulation that would prevent this accumulation here. And this will happen. This can be happen by having an energy barrier. And what is interesting is that this energy barrier depends also on the PE to PC ratio. And just to finish, that I see I have like 1 minute or 2 minutes left. Let me just show you two illustrations that are in agreement with our hypothesis. So first of all, there is this enzyme called DGA1, so in yeast. So it's the mammalian equivalent of DGAT2. So when lipid droplet forms, DGA1 moves to the lipid droplet surface. And there was a paper showing that it irreversibly moves between the ER to the lipid droplets, which was puzzling to us because we found, based on our data, that it would prefer staying on the droplet. But there is this nice study showing that actually this retrieval from the lipid droplets is mediated by AC2. So without AC2, DGA1 stays at the monolayer, which is concordant with our model. So basically, to retrieve proteins from the droplets, you need to put some energy because the system is at the lower stage. So if you want to bring it to the bilayer, you need to combat this energy. So with this, I want again to thank the organizers for having me in this great symposium. And I want to thank Lucie Caillon, who's presenting most of the work I presented you today, and Emeric Charlet, who's done the work on the anthropathic helices. I want to thank all my collaborators and of course, my funding agencies. Thank you for your attention.
So, thank you very much, Rachid. It was great to see all those techniques that you use to study lipid droplets and their dynamics. So, I'm going to go through the questions. The first question is mine. So, have you explored how specific determinants on the structure or sequence of a protein domains affect the affinity towards the composition of the hydrophobic core of the lipid droplets?
Yeah, I think the easiest answer is to say that's the next step. I think what we've tried to do first is just identify with the minimal peptide how the partitioning is affected. And clearly, if you change, based on the early data I showed on genuine proteins, you see that the partitioning degree differs, and that suggests that the sequence itself matters. And one short answer also is, based also on recent findings on the hairpin of DGAT1 of DGAT of GPAT4, is that if you would simply add a charge within the hydrophobic region, you it would be unfavorable for this charge to be in a dielectric medium, which is the oil phase. So that can explain why you have some structural changes to put the charge at the interface, so as to minimize the contact with the oil phase. So in a nutshell, I think there are many features, amino acid features that can impact the partitioning.
Thank you. So next question is from Barbara. Does the length of the transmembrane domain play a role in partitioning between the bi or the monolayer, or
Yeah.
is this a factor neglectable due to the lipid phase inside lipid droplets?
Yeah, that's a very good question, and we thought initially that would play. So I showed you the data where we have these 20 amino acids. So we had longer. So 20 amino acid match roughly the thickness of the ER, and we've worked with 28 amino acids. So because the driving force is so strong, we could not see any difference between 28 and 20. But to some degree, when you start relocalizing the protein in the bilayer, when you have a lot of PC and P, then you can start seeing differences that are tiny. But we don't want to pronounce on it. To engage ourselves on it, because we need a deeper study to really fine tune those differences.
So next question is from Maria. Is the lower phospholipid density in lipid droplets due to their higher curvature, that is the bending generated spaces between the phospholipids?
That's an interesting question also. So I doubt it's due to the bending of the lipid droplets. And what we think, and we have data supporting that, is, when you have a bilayer, you have forces that are sticking the two monolayers together. So that's called the adhesion force. So you have two monolayers that are adhering to each other, and this has some energy. On a lipid droplet, you don't have those energy because you have just one monolayer. So the fact you have an adhesion forces on the bilayer, that tightens up the phospholipid bilayer, and on the contrary to the monolayer. That's what we think is the gist for this difference between the monolayer and the bilayer packing.
Okay, then a question from Zeba. Did you check how other lipids, for example, cholesterol or unsaturated fatty acid side chains, change the preference in the monolayer versus the bilayer?
Yep. It's reflecting the meeting. I'm just having great questions, like the meeting. So, yes. So historically, you as you can appreciate, the readout was so black and white that we put into doubt ourselves, because for a year we've been saying, okay, these are like trash. After we start understanding that the phospholipids play a role. So if you use PA, which has the same curvature as PE, you have the similar trend as PE. And if you change the oil phase, also you use squalene, you change all this stuff. And definitely my gut feeling is, of course, if you change saturation, all these parameters, they would change. But we didn't have time to prospect all those parameters yet.
Okay. So a question from Rashi: are these bulged lipids, lipid drop, always just more spaced lipids, or can they have more lipid molecules?
Can they have more lipid molecules together? I'm not sure I get the question. Are these bulge lipid droplets?
If it's not clear, maybe we can ask Rashi to make it a bit more clear, and we can move on to the next question. So what is the content of an ER lipid droplet in the cell?
So in terms of lipids, of course, there seems to be. I mean, it's not clear yet whether what seems to be clear is they have so lipid droplets originate from the ER, but seem to have a different phospholipid composition. So therefore there might be some phospholipid editing that favors the recruitment of specific phospholipid to the lipid droplets. So they don't have the same phospholipid composition for sure, and there are like all the data showing that you have some asymmetry on the ER phospholipid composition that needs to be carefully redone, I guess. But since lipid droplets emerge from the site from the ER to the cytosol, you could expect that lipid droplets would have phospholipidome reflecting more the cytosolic phospholipidome of the ER. But these are pure speculation.
We have some more questions. If you don't mind, we can continue for some time. So, does the accumulation of lipid droplets affect the normal events in a liver cell?
Yeah, I mean, I'm not the best person to answer on that, but during steatosis, you have abnormal accumulation of lipid droplets and so on and so forth. But what is interesting though is there is a specific liver steatosis triggered by an imbalance in PCPE, right? So if you increase the PE to PC ratio, you get the steatosis. And this is interesting from my standpoint, because if you shift the PE, if you increase the PE to PC ratio of the ER, what would tend to happen is you would just like drive more proteins from the ER to the lipid droplets. And that might be actually a signature or cause of steatosis. But this, I guess, needs to be further prospected.
Great. And one last question. So, how are proteins residing in lipid droplets degraded?
I think that's the question for Pedro, right? So I can cite the review papers that I've read. So basically, you have a the CMA, so chaperone mediated autophagy. So basically, it's an autophagic pathway by which you ubiquitinate the proteins at the lipid droplet surface. They are recognized and then degraded through the liposomes, through the lysosomes, and possibly also you have lipophagy, basically whereby the autophagosome basically just engulfs the lipid droplets and degrade it also through the lysosomal pathway. So I think and maybe Pedro can wade in if he's interested. Yeah.
Okay, so I really want to ask a question, so maybe I can close the session with this. I saw that you are able to reconstitute lipid droplets on GUVs. Have you tried isolating lipid droplets from the cell, containing all the lipid droplet associated proteins, reconstituted and study how difference in the mobility of proteins from the monolayer to the bilayer?
That's a so we haven't probed the diffusivity, but we have reconstituted already genuine lipid droplet proteins. So during my talk I showed some examples, but we've studied the diffusivity at the lipid droplet surface. of many lipid droplet proteins, and we haven't like really deeply uh, done experiments to address the question you just asked, but that's something interesting to do in the future, of course. Yeah.
Okay, then I would like to thank you very much
Thank
for your
you.
talk and your question and answer section.
Thank you.
Uh, and then now we have our session break until until 3 p.m., where we're going to talk about viruses with Eugene Koonin. So thank you all for being here.
Thank you.